Okay, so we need to look at operations and functions of the cell. That means I'm going to go into a little bit more detail about what each organelle does. Okay, and then we're also going to look at how the cell transports material, both intracellular transport, that means once it's in the cell, how does it move around, and a couple of ways that it gets in and out of the cell. So transport across the membrane as well as just within the cell, right? Because those are a little bit different. They sometimes require different processes. Okay, so we need to understand the significance of this big word, compartmentalization. Compartmentalization is crucially important. It is what makes the kind of cell we just labeled, which is a eukaryotic cell, okay, both plant and animal, okay, that is a cell that has organelles, okay, separate compartments. That's where compartmentalization comes from a prokaryotic cell, like a bacterium that doesn't have these compartments. Okay, having the compartments and having compartmentalization makes a cell far more efficient, right? Because it's essentially like having a separate room for each job where the conditions for that job are ideal. Okay, and we'll talk about an example of that in a little bit. We need to know the role of each organelle. All right, so that's just something you're going to have to study and memorize. Understand that cells are a complex system of interdependent processes. Okay, interdependent means they depend on one another, right? If one of the links in the chain breaks, the whole chain is useless. So we got to make sure that all the processes are working. Yes. There will always be more memorization in biology than in the other sciences, but you'll also be asked to apply a lot of the stuff that you have to memorize. Okay, number 4, understand how the cell carries out its functions. So basically that's kind of like number 2. Those two kind of go hand in hand. Why does it have to have a smooth ER and things like that? Okay, and fifthly, understand how materials are transported across the cell membrane and within the cell. So intracellular means within the cell, and then we've got transport across the membrane. There's many processes that are involved with that, and it mostly depends on the size of the molecule. Okay, compartmentalization. So cells are divided into compartments by membranes. Each organelle has its own membrane that kind of makes that little bubble that is that organelle. Okay, and that's important because a lot of the processes that go on in the cell are not compatible with one another. For example, the digestion of food requires an acidic pH and enzymes that are designed to break down cellular material. Those are not two things that we want to have in the rest of the cell. Agreed? Because, well, that would eat the cell. So it's good to have a separate environment where food is digested. The same goes for an environment where sugar is broken down. The process of breaking down sugar releases a lot of heat, and heat would be harmful to the rest of the cell. So we want to have a separate double-layered compartment that insulates the rest of the cell from that process. Okay, so we have these different compartments that allow those processes to go on under their ideal conditions and keeping them separate from the rest of the cell. That makes them far more efficient. Okay, so a eukaryotic cell like this one, okay, and eukaryotic is a term you will need to know. Okay, eukaryotic cells are cells like ours. They have a nucleus and organelles. Okay, a eukaryotic cell is like your house. Within your house, there's the environment in which you live. That's like 21 degrees. And then there's your refrigerator, which is probably 4 degrees. And then there's your freezer, which is like minus 15. Okay, those 3 separate environments each do a different job. If I didn't have those 3 separate environments and I wanted to balance the 3 jobs, that is, make me comfortable, keep my food preserved for over the short term, keep my food preserved over the long term. If I wanted to do all of those jobs in the same environment, would I do any of them very well? No, it would be like, okay, to do all these jobs, I have to keep my house at 10 degrees. So I'm cold, my food spoils, and I have no ability to store food over the long term. Okay, finding a balance between jobs that are not similar is difficult. And if you do find that balance, you're not doing any of them very well. It's like being a jack of all trades, but a master of none. Okay, sure, I can do all of these things, but I'm not very good at any of them. All right, I'm not specialized enough. Okay, so compartmentalization provides different local environments, okay, to allow the cell to perform metabolic functions that aren't compatible. Okay, if you are a bacterium and you don't have these organelles, you have to do that. You have to perform all of these functions in the same area. Okay, this is why you never see a multicellular bacteria. Okay, bacteria are strictly single-celled. They're quite primitive, and they're extremely small. Okay? They're just not efficient enough to grow beyond a certain size. Okay? All the jobs have to be done in the same place, as opposed to a eukaryotic cell where jobs are all done separately. Now, a eukaryotic cell having all these organelles also means eukaryotes can become multicellular because they can become specialized. And that was what we were talking about a minute ago. Muscle cells would have way more mitochondria, where liver cells would have way more smooth ER. Okay? You don't see that in a bacterium because it doesn't have any of those things. So you never have to worry about a multicellular salmonella organism coming and eating you. Yeah, because they're just not going to do that.
Where cells are what are bacteria?
Prokaryotic. Prokaryotic. So instead of the EUK, it's PRO. Or sorry, instead of EU, it's PRO. So prokaryotic or eukaryotic. Okay, questions there on what compartmentalization is? As long as we understand what it does. It creates separate environments to make processes more efficient. Essentially, that's what it boils down to. Okay, so compartmentalization allowed cells to become larger. So here's prokaryotes. Okay. Prokaryotes have no compartments and are, as a result, much smaller and the eukaryotes are more specialized in their functioning. Okay. So here's give you a kind of a size example. Here's an animal cell, and we can't see the whole cell because it's too big. Here's a bacterium, right? Scale 0.5 micrometers, right? You wouldn't even have seen that yesterday using the microscopes, right? The smallest thing you looked at was the euglena, and it might have been 60 micrometers, okay? So a virus is pretty small, okay? A bacterium is maybe only 2 to 5 microns at the biggest, okay, for a bacterial cell, whereas an animal cell is huge by comparison. Yeah, viruses, they've resolved them with electron microscopes. Basically, you can't see them with a light microscope. A micrometer is a micron, sorry. Yeah, micrometer. All right, so that gives you some idea of the size. And you can see kind of within the bacterium here, here's the DNA. See all this snarled stuff? That's the DNA. Notice there's no nucleus to contain it. All right, so the bacterium's DNA is in there with the digestive enzymes and with the cellular respiration and all that going on. So can the DNA of a bacterium get damaged? Yeah, that's why bacteria mutate so often. Okay. Simpler organisms, especially prokaryotes, are easily mutated. All right, doesn't mean they're going to die. It also doesn't mean they're going to, grow giant claws out of their hands or shoot webs out of their fingers or whatever. Okay, that's like all science fiction. Okay, but they mutate slightly, and that may do something like make them immune to penicillin. Okay, when I was in university, one of the labs we did was we took E. coli bacteria, okay, which can cause pretty serious food poisoning incidents. Okay. And we put it under an ultraviolet light for 30 seconds. Then we smeared it on Petri dishes. One Petri dish was just nutrients. The other one was nutrients with penicillin. Okay, and what we found was that bacteria that hadn't been exposed to the UV all died on the penicillin agar plates. Okay, but the ones that had been exposed lived. So what did we do? 30 seconds of exposure to ultraviolet light, I created penicillin-resistant bacteria. Okay, so they can mutate pretty easily because they're simple. Okay, but being simple also sometimes makes them more resilient. If something goes wrong with one part of an animal cell or a plant cell, remember, we're looking at all these interdependent processes. If one of them goes wrong, the cell will die. A bacterium tends to be far more resilient. We can find them in environments where no eukaryote would ever live. Okay, we find them in like the the geysers and the stuff like that in like Yellowstone Park in extremely hot, extremely basic, extremely acidic environments, and we find bacteria living in there where no eukaryote would possibly survive.
Is that why they can repeatedly, because they
Yes.
change?
Same with a virus. A virus can easily mutate as well, right? So, I mean, I'm one of those people. I believe in vaccinations. And if you're an anti-vaxxer, that's too bad for you. I don't care. But if I'm one of those people who believes in that, but at the same time, I never get a flu shot. Okay, because the flu virus mutates so often that a flu shot is really just everybody's best guess as to what strains are going to be around this year. Most likely it would work and it would prevent you from getting terribly sick. But so does getting sick one time and then getting better. I get the immunity too, right? So I got a flu shot once and I've never been sicker. The one time I got it, I was like crippled sick for like 3 days. So I never got another one. But yeah, that's how that works, right?
But eukaryotic cells are also found in really harsh environments like Antarctica, stuff like that. Wouldn't that be
Not eukaryotic cells in Antarctica. Oh, okay, but they're a multicellular organism. They may not necessarily have evolved there.
So what you're saying is that if you have like a piece of meat that's infected with E. coli, if you brought it out into the sun for a while, like if you
No,
vacuumed
don't.
the sun,
No. It might kill the bacteria, but you got to remember that the toxins the bacteria have produced are still in the meat.
so it would make it worse if you had the meat sun rays, because the UV could mutate it to make it.
Oh, no, it would be nothing like that. Like, I mean, we put just bacteria that we had grown right under it, like just a pure black light for 30 seconds. Right? Like if I had to put my hand underneath that black light for 30 seconds, I'd come out with a sunburn. Like, yeah.
just double checking. So are
Prokaryotes, yeah.
are they like a type of cell?
Yes, prokaryotes are a type of cell. So a virus is not a prokaryote. A virus isn't either, right? Because it doesn't have cells. Bacteria, blue-green algae, and simple things like that are prokaryotes. An animal cell is always going to be eukaryotic. Always. Yeah.
Why don't viruses have cells?
They just don't. They are simply genetic material encased in protein. Yeah. No one's really sure on that one. Because they're so small, the lineage of viruses is hard to trace in the fossil record, right? It's hard to find them. Oh, thanks. Thanks.
The reason that flu shots work is because in other countries, the flu will mutate there first, and then mutate almost the exact same over here. So they find out what it's like over there, find a way to treat it, and then bring it to here before it even mutates.
Well, that's what they would hope, yes. But
It doesn't always work, but that's how they do it.
like I say, it's their best guess as to what's going to be around for a strain each year, right? Now, I'm not saying don't go get a flu shot. Go get a flu shot. Absolutely, right? Vaccination is the single biggest medical advance in the last 200 years. Okay, it's huge. There's a reason why we do it. But I'm saying, like, for something like the flu, unless you're, let's say, like elderly or very young, then where the flu could be dangerous or possibly fatal. Right? I mean, we're all in our physical prime. We're highly unlikely to die from the flu, right? I mean, we'll get sick, it'll be unpleasant, but what do you do? Yes. Okay. So DNA is used to make materials. So basically now we're talking about, here's some functions. So within the nucleus, that's where all the DNA is. Okay, so DNA is used to make stuff. So within the nucleus, okay, is all of your DNA. Inside the nucleus, the DNA is all unraveled. It's like if someone took a ball of yarn, completely unraveled it, pushed it into like a lump and just chucked it. That's what the DNA looks like inside the nucleus. Okay. When the cell's dividing, it all kind of winds together and makes the chromosomes. Okay, but when it's in the nucleus in its active form, it's all unraveled. So imagine trying to be the nucleolus. Someone tells you, hey, go find the part of the DNA that codes for this. It would be like going into a hoarder's house and trying to find one item in particular. Okay, like it's amazing that it ever works. Okay, but that's what kind of it looks like. Now, the nucleus itself, under an electron microscope, this is what the surface of the nucleus looks like. So we're seeing basically just the nuclear membrane here on the right. Okay, you can see kind of these bumps. The bumps are what we call nuclear pores. They're the only way things get in or out of the nucleus, is through these little gates, okay, these pores, right? And basically the only thing that goes in or out of there is DNA or copied DNA, which is RNA, that is used to make the proteins. Okay, zoomed in on those little pores, that's what they look like there. And then zoomed in even further, okay, we can see kind of the interlocking series of fat molecules that kind of make up the membrane itself.
How do they copy DNA?
There's a thing called DNA transcriptase, which is an enzyme that the nucleolus produces that unzips the strands and copies one side. Okay, because the sides of a DNA molecule are complementary. Right, you guys remember that from grade 9, right? Like A always goes with T and C always goes with G. Okay, so it only needs to make a copy of one side to know what the sequence is. So it'll just unzip it, copy one side, and put it back together. All right, so from the nucleus, that's where protein synthesis happens. The nucleolus, actually, in particular, is the part that does the protein synthesis or controls the protein synthesis. Okay? The nucleus is just where all the DNA is. All right, this is how protein synthesis works. Now, I've already explained it to you once, and I said that basically all you need to know about protein synthesis is this. The nucleolus copies the part of the DNA that has the instructions get sent to the ribosome makes the proteins. That's the extent to which you need to know protein synthesis. Nucleolus, copy DNA, ribosomes make protein. That's it. Okay, this is a far more in-depth kind of a thing. Like you were asking, Jacob, how does this work? Okay, so you got DNA transcriptase, RNA transcriptase, which pulls the DNA apart. Okay, makes a copy of one side. That's this pink ribbon here. Okay, so the pink ribbon is the copied DNA. It goes out through one of the nuclear pores. Okay, so you can see that right here. And then it goes to a ribosome. Okay, so this is a ribosome here, which can then read the DNA, and it starts acquiring all the pieces that it needs. So things will bring in those amino acids, that's what makes up a protein, and it'll start stringing them together in the right order to build a protein. Okay, when that's done, the RNA is either sent back to the nucleus or is simply broken down by the cell.
Isn't RNA much more easily modified as well?
Well, it's only one strand, right? It's not so much that it's more easily modified, it's just single-stranded DNA.
I saw a thing that had said like
Octopi is the plural of octopus.
but also they yeah.
No, well, you looked it up wrong. Hippopotamus, hippopotami, octopus, octopi. And the ends in us, ends in I as a plural. That's how the English language works.
Well, I heard they were most of their stuff was RNA versus DNA or somehow.
No, an octopus is a multicellular eukaryotic organism. It would have double-stranded DNA in the nucleus of all its cells. That's there must have been a misunderstanding there of some kind, but that's how that works.
It was a lot easier for them to take different ways.
Okay. Yeah, I don't know what to tell you. Okay. So like we said, that's the extent. You don't need to know even as much as is here. Nucleolus copies the DNA, or the RNA goes out, and proteins are made by the ribosomes. Okay, endoplasmic reticulum. Sorry, Jake.
Is there any risk in transporting that DNA strand? Could it
No.
possibly get mutated on the way?
I suppose it's possible, but then all that would happen was the wrong protein would get made.
Can that have an effect on the cell?
Not really, no, because we're only talking about one ribosome, right, making the wrong protein. Not a big deal, right? The message would be copied and sent out to many ribosomes. There's millions of them, right? So we're making lots and lots of that protein, and it's gonna, yeah, naturally a few of them are gonna have mistakes, right? I mean, it's like, if you got a factory that's building cars, sometimes a car comes out and something wasn't done quite right, breaks down. Okay, so much of the membrane volume of the cell, so within the cell, is endoplasmic reticulum, both smooth and rough. Okay, under an electron microscope, this is what they look like. Okay, so we can see that on the right-hand side here is all the rough endoplasmic reticulum. And it tends to fold on itself in a fairly predictable pattern, right? And we can see the ribosomes here are stuck on the outside of that ER. But the smooth ER, which is here on the left, okay, is just everywhere, right? There's no kind of pattern to it. It just folds all over the place and fills up a lot of the volume of the cell. That's important because it does so many jobs, right? So they do look a bit different, but in general, they're still tubes, okay, or folds and tubes that are doing a job. Okay, so within the smooth ER, okay, so within the membranes of the smooth ER, lipids are produced. Okay, that's fats, and that's used for building or repairing membranes. Okay, complex carbohydrates get broken down, so that's starches, and poisons are detoxified. Okay, that's the smooth. And also hormones. Some hormones get produced in the smooth ER. Some, not all. Okay. Basically, though, all of these things are things that need to get out of a cell or to the outside of the cell quickly, right? And the smooth ER has access to a lot of that. Okay. What did we say the job of the rough ER was? It had a much simpler job. That's the Golgi. It's covered in ribosomes, and ribosomes make protein. So the smooth ER, or sorry, the rough ER transports the proteins. Okay, just helps them to get around faster than they would by diffusion or anything else. Okay, lipids can be moved quickly to needed sites, okay, basically on the membrane, wherever the membrane's broken or damaged, okay. Poisons and drugs are detoxified quickly and removed from the cell to the membrane, okay. Travel through the ER also limits contact with the rest of the cell. So if I have a toxic material and it's in the smooth ER, it's only damaging the smooth ER and not everything else. If it was just allowed to, kind of wander throughout the cell, it would damage a lot more stuff. Being enclosed in the smooth ER kind of isolates it a bit more. Okay, the Golgi apparatus. That really, to each individual cell, not much. In general, your non-steroidal anti-inflammatories, so Tylenol, ibuprofen, like naproxen, stuff like that. Basically, they allow the fluid that results from swelling to go back into the bloodstream, and so it helps to reduce swelling. Reducing swelling tends to reduce pain. Okay, so if you have a bad headache, it's thinning the blood a little bit because fluid is moving into the blood. That reduces your blood pressure a little bit, and that can relieve pain and headaches, stuff like that. Yeah. Okay, now for the Golgi apparatus, okay, it's important. This is what everyone draws it to look like. This is what it actually looks like under an electron microscope. Okay, it's a bunch of folded layers of membrane. Okay, but there's two sides of it. One side of it is the receiving side, and the other side is the shipping side. Okay, so the stuff it needs to get rid of always comes in on the same side. And the stuff that it's getting rid of always goes out on the other side. Okay, so it's called the cis face and the trans face. Okay, the cis face is the receiving side, and the trans face is the shipping side. Okay, so you can see that this here is the receiving side of the Golgi. So stuff that's small, a few kind of small vesicles are coming in on this side, but you see on the other side, there's lots of large vesicles. These large vesicles are what the Golgi produces. In fact, this is one being produced by the Golgi apparatus right now. Okay, and they go out and they go very quickly to the membrane. The vesicles are made out of membrane material. So as soon as they get to the membrane, they just merge with the membrane, and whatever was in them pops out of the cell, okay? And then it just kind of rejoins the membrane.
So is it kind of like a whole bunch like folded paper, or is it like
more like tubes, folded tubes. Like if you took like surgical tubing and just folded it back and forth, Yeah. No, I'm never going to ask you a cis face, trans face. No. Well, it actually, its membrane isn't always getting bigger because the same process happens when cells take stuff in. right? They engulf it, and then that vesicle moves into the cell. So the membrane is constantly being added and taken as stuff moves in and moves out. Yeah. I mean, cells do grow, but it's not necessarily because the Golgi is always pumping stuff to the membrane.
So the receiving end of the Golgi can also receive
Small ones, yeah. Most of the time the stuff that comes to the Golgi isn't packaged, but sometimes it is. It depends on the cell. It's completely dependent on the cell. Muscle cells often, nerve cells never. So it all depends on what they're doing. Okay, lysosomes. Okay, so we kind of talked about what they do the other day. They're responsible for digesting food. So here's that process, Jake, that you were asking about. Okay, if a food particle encounters the membrane folds around it. Okay, and then that vesicle will come in. It's called a food vacuole if it contains food. Okay, and it'll meet up with, and this is actually wrong here. Okay, this is a peroxisome. Okay, the peroxisome meets up with the food vacuole. This is a lysosome. Okay, so we have the Golgi actually making these digestive enzymes, okay, and pumping out the peroxisomes, which then become the lysosomes. Digestion occurs. Okay, it's also how a damaged or malfunctioning organelle is recycled. To your cell, everything is food. Right? So if you've got, and like in this case, a mitochondria that's damaged, not functioning anymore, it'll just eat it. Okay, and well, we might as well get something out of it. We can recycle the materials. Okay, so your cell will break it down and recycle the materials, make something else.
like why can't I do that? Counter something. Use that.
Because it won't do that to your DNA. Like it would have to go into the nucleus. It's not going to go in and digest the nucleus, right? Because then the cell will die. I mean, when a cell is killing itself, that's exactly what it does, right? It just releases that stuff everywhere and the cell dies. But all right. So what would you write on a test if I asked you what the function of the lysosome is? Digest food, digestion. Yeah, something like that. Like just a quick answer that's descriptive enough. All right, the mitochondria. So the mitochondria under an electron microscope looks a lot like what we draw it. And it's one of those few things that actually looks kind of like what it appears in pictures. Okay, so we got these folds on the inside. The purpose of folds, and because you see that a lot in living organisms and in cells, is to increase the surface area. Okay, if I just had this mitochondria and it was just shaped like this, I would only have the interior surface area for reactions to take place on. But if I make the inside of it folded like this, have I just made a lot more surface area on the inside? Yeah. Okay, so it's increased the surface area. The inside of your small intestine looks the same. Okay, you got these things called villi, which are little tiny folds. Okay, if we like took the intestine, not that we would do this, but took the intestines out of a person, okay, and we were to spread them out, there's enough surface area in your intestines to cover a tennis court. Okay, if we're one cell layer thick with all the folds and everything, that's why it can absorb so much of your food energy so quickly.
How do we figure out what's inside of these things we cut?
Well, the electron microscope has basically done a lot of that.
So they cut, like they cut a cell in half.
No, it's more that they coat it. I mean, yes, they are in some cases cutting a sample and then coating it with like a scanning electron microscope coats everything in a fine layer of gold and then shoots the electrons out and they reflect off. It's a very expensive process. But yeah, that's basically what we've been doing. The electron microscope, once you look at something with an electron microscope, it's dead. The process of preparing it for the electron microscope and then bombarding it with electrons kills it. Okay, but yeah, that's essentially what we're doing. We're looking at the inside of them. I mean, with a light microscope, we could actually see, if we had a good one, a lot of these details as well. Right? It's just an electron microscope can resolve it much more. All right, so like we said, within here, okay, within the mitochondria, we're carrying out the burning of sugar. Essentially, cellular respiration is going on in here. Okay, the breaking down of sugar to release its energy goes on within the mitochondria. Okay? So if you have a fever, these things are working overtime and driving your body's temperature up.
So when you give an exhale, we exhale carbon dioxide, does that come from the
Yep, that's exactly where it comes from. Okay, so yeah, within all of your cells, that process is going on. The carbon dioxide diffuses out, the oxygen diffuses back in. The carbon dioxide goes to your blood goes to your lungs, okay, and it diffuses out of your blood into the air in your lungs, and you exhale it. Okay, and then the chloroplast. Okay, obviously you're going to write basically one word for the function of this, which is photosynthesis, right? Making of food for the plant cell. And they're, like we said, they're small, but they're not that small. Okay, they're probably a good 3 to 5 micrometers across, which is why when we do the cell anatomy lab, you'll actually be able to see them. Okay, within the cells of the leaf that we look at, they'll be kind of grayish green, and they'll have essentially this same shape. Okay, they'll be kind of flat on the bottom and round on the top. The thylakoids, no, you won't see those. You'll see these kind of green bubbles, and that's what you'll see. Okay. Yeah, they do look like stacks of coins. I think I said that in the podcast, didn't I? Stacks of coins. I know I said in period 3 and 4. But. Okay, and then it also has that double membrane layer. All right, and then the other stuff here, and this is some pictures of some of the other things here, but you guys were asking about the cell membrane, right? Like in all of the pictures, it looks like this smooth layer, but this is what it actually looks like. Okay, the cell membrane is, yes, a whole bunch of fats that are turned side to side, okay, but there's also glycoproteins, which is a sugar and a protein put together. There's receptor molecules. Like the whole thing is actually far more detailed and intricate and quite chaotic, actually, okay, than we often represent it to be. Yeah, they can move around a bit inside. Yeah, not that they actively move, but yeah, they kind of. Yeah. All right, so yeah, the cell membrane's quite a bit different than we had imagined. Okay, and these are the 2 words you're going to need to use when you tell me the function of the cell membrane: endocytosis and exocytosis. Endocytosis is movement into the cell. Exocytosis is movement out of the cell. Right? The cell membrane controls those 2 things. Okay, and on a quiz or a test, those are the words you need to use. Don't say controls what gets in and out. Controls endocytosis and exocytosis. Be technical.